Johnson and Sheffers and a number of people have all documented this same thing. Jessica Sheffer and her PhD thesis at Purdue University 2 years ago said, well, if that's really the case, and she demonstrated that it was, you should be able to nullify the herbicidal activity of glyphosate by merely adding a fungicide. Added Ridamil, inoculated with Phytophthora and Pythium, no herbicidal activity of glyphosate, and some very susceptible varieties otherwise. So she looked at glyphosate resistant, Roundup resistant weeds, and she found that it didn't matter if she added the fungicide or not, they were still resistant because they're resistant to the fungi, not to the chemical. When we dump on 2 and 3 and 15 times as much herbicide and we'll get a little better kill, it's not because we're killing the plant with the chemical, it's that we're increasing the virulence of the pathogen and we're destroying the natural biological controls that would suppress those pathogens. As a consequence, we now have over 40 diseases that we used to consider relatively well managed and controlled in our production agriculture. I've served for 40 years on our Threat Pathogens Committee. We now refer to these diseases as re-emerging diseases because our management program, whether it was with resistance or whether it was with cultural practices and nutrition, aren't working as well because we have super pathogens and super weeds. And it's those super pathogens then that affect other crops in the rotation. They're not pathogens just for one crop. Your soil-borne pathogens can have a very wide host range. And then when you change the virulence with glyphosate, we find that the host range also expands so that they're not specific in the same categories as they used to be. We see the other factor with the increase in our toxins in our food. Fusarium head blight, the fusarium toxins, aileron, deoxynivalenol, the nivalenol, and even now the T2 toxin that was part of the yellow rain that was used by the Cambodians, North Vietnamese in an attempt for mass genocide against the Hmong people. That toxin had never been produced in our environment until we had the genetically engineered Roundup Ready plants, and we now see the T2 toxin predominant in many of our cereal products. The representatives from the baking industry at our National Fusarium Head Blight Forum 2 years ago said we can no longer obtain enough domestically grown grains to meet their needs. They have to import it because of the high levels of the toxins. Andreas Tiedemann reported at that meeting that it's not just in the grain now, and with the head infection, you can have a plant in the grain that looks very healthy. But the toxins now, since Fusarium is a root colonizer initially, the toxins are also produced in the roots and then translocated up so that it's not a matter of just turning the fan up on your combine and blowing out those high toxin kernels because they're very lightweight, because the toxins are translocated up and they're accumulating in the grain. Had others that reported that it's no longer safe to use corn stalks and wheat straw and barley straw as bedding for your animals because it can leave them sterile from the skin absorption of the zearalenone, an estrogenic toxin produced by the fusarium. What Dr. Fernandez found in Canada in her 8-year study was that if you have used one application of glyphosate as a burn down, in the previous 3 years, you'll have a significant increase in head scab and the toxins. If you use it every year, it's about a 300% increase. If you're going to grow your cereals after Roundup Ready canola, it goes up exponentially because you're applying more glyphosate. It's going through that root system of the canola plant, so you're changing more of the soil biology that would help to minimize those pathogens. Again, there's nothing in the Roundup Ready plant that does anything for the glyphosate. It's still a very strong chelator. In this study by Zobioli again, University of Missouri, you see the effect of glyphosate when it's applied on a Roundup Ready soybean plant. You see the reduction in lignin. You'd expect that because that's through the shikimate pathway, and the Agrobacterium tumefaciens pathway is not as efficient as the normal one, so that you're going to have reduced lignin. But with the glyphosate as a strong chelating agent, you're also immobilizing those nutrients that the plant would need in order to produce that lignin. And so there's less lignin. That's a structural component. A little harder to digest for dairy, so a lot of people said, hey, maybe that's a real plus for us, until we did some of the analysis on that lignin, and it's the type that is even more difficult to digest. You look at the nutrient density down here with the amino acids. And if you want a reference point, those red lines are about a pound of glyphosate per acre, about the old recommended herbicidal rate, and you see reduced amino acid density in that crop. It's less nutrient density. You look up at the top, you see less efficient photosynthesis. How are you going to capture the sun's energy as efficiently and store it as sugar if you don't have those nutrients available, if you don't have the physiology functioning? The other thing that you see is that it takes twice as much water to produce a pound of dry matter in a Roundup Ready plant treated with glyphosate than in its isogenic non-Roundup Ready parent. How are you going to solve the problems with increased water shortage or drought stress or increasing temperatures if you've reduced the water use efficiency by 50%? And the more you use, the greater that drops. Now you can say, well, is that really significant? Well, I can tell you in Iowa and Nebraska and the Midwest in 2012, 2013, it was a very serious problem. For my neighbors, they pay about $100 an acre for their electricity for their irrigation water. It's not just that we're concerned about how much water, but also the expense and overhead that's added to that production system. You can see these two soybean fields, Nebraska, 2012. You have the Roundup Ready beans that receive 2 applications of glyphosate because you have a lot of late emerging weeds now coming up that weren't killed the first time. And then you look across the fence row and you can see the non-GMO, and they're much greater water use efficiency and drought tolerance. Same thing for corn. These two fields were separated by a 60-foot gravel road. At 100 bushel difference in yield. Yeah, you need water for both of them because water provides the hydrogen that's combined with the carbon dioxide from the air to form the sugar. That's where we get our oxygen. Manganese is critical in that process. As Dr. Barrett, Professor Barrett at Cornell had a little picture of of a Buddha god behind his desk, and he had all of the world's population bowing to it, because he said if it wasn't for manganese, none of us would be here. And he had called that little Buddha god manganese up there, because that's where our oxygen comes from. But the plants get their hydrogen from that water. You can see that stress and its impact. We have the resistant weeds. This was the whole purpose for genetically engineering herbicide tolerance. We now have over 60 weeds that are resistant to glyphosate. In the southern states, pretty well nullified the technology. We're saying the same thing. It's just a matter of time before it moves across. And it starts usually with a few resistant weeds. You can see them down the front there with the pigweed in this field in Georgia. 3 years later, this is the same field on the right. And that's why a farmer has to have a GPS unit to find his corn crop in there. The weeds are able to take over. It's a terrible problem because they're much more resistant to the, not just the Roundup program, but all of our other control systems aren't nearly as effective now because they're resistant to the pathogens. They're more tolerant to our control mechanisms. But the food and feed safety concerns are very well documented now. It's taken a while. We have very limited research from a long-term basis because of the scientific censorship that's gone on and the repression of data. These are some of the areas that we know are of concern from a feed safety, food safety concern here. I'll get into some of those. I've mentioned the lower nutrient density of our foods. Say we have an obese problem. What happens in obesity is the body's taking in more than it can digest and utilize. It wants to get it out of the way. If it doesn't have those micronutrients to keep the physiology functioning, it stores all of that carbohydrate as fat, accumulates around the waist. It's probably as much a micronutrient deficiency as any other factor. And when you look at the reduced nutrient density on many of our foods, our genetically engineered foods, when you look at just sulfur alone, methionine is a starting point for all our proteins. You got a 52% reduction in Roundup Ready alfalfa compared to the non-Roundup. Look at your manganese for liver function, for resistance. 80% of the livers in some of our commercial slaughterhouses have to be discarded, disposed of because they're in such terrible shape that they're not considered even fit for pet food. Dr. Art Dunham, a few years ago, convinced Iowa State's large animal lab to install an ICP unit to measure and analyze the livers and other organs that come in with all of their animals that are necropsied. In other words, all of their diseased animals. 100% of them the last 2 years are extremely deficient in manganese. Jeffrey Sheffords, Minnesota veterinary toxicologist, showed that the reason for their stillbirths in their cattle and their deformities, that 100% of them are deficient in manganese.